All right, guys. Hi, how's it going? Snow day again. So today we had on the schedule to start the third section of notes on variations on Mendel. So we're going to do that still at least in part today. So the other thing is I will send an attachment of this PowerPoint to you guys in the email that I send. But also be taking notes just as if you would in class. We will maybe touch base on some of these things, might be questions on some of these tomorrow. We're going to go through the first three things on this list here that you're seeing on the screen. And then as a note, the corn activity that was due today and the genetics packet number 1 that was due today will obviously just make that due tomorrow. So here we go. So we're going to talk a little bit about some variations on Mendel today. So basic Mendelian genetics, like we've been talking about, was this idea of simple dominance versus recessiveness in terms of the alleles that were passed along from parent to offspring. The whole fact was that you needed just one copy of the dominant allele in order for the dominant form of that trait to appear within the offspring. Now, in some variations on this, it's not quite that simple in some cases. And we're going to go through each one of these 6 here: sex-linked traits, codominance, incomplete dominance, polygenic traits, pleiotropy, and then we'll talk about genes and the environment and the role that environment plays on influencing phenotypic outcome. So the first on this list is sex-linked traits. Let me flip to the next screen here. We talked about humans have 23 homologous chromosomes, right? Or 46 total chromosomes, but 23 pairs, I should say, is what I should say. We inherit 23 from mom and 23 from dad. Of these pairs, 22 of the pairs are called autosomes that do not determine our gender. So they contain genes that ultimately determine all traits minus gender. There's one pair of chromosomes called the sex chromosomes that are responsible for determining gender. And there's my son Anders sneaking through the background. That one pair, they're called the sex chromosomes. They're responsible for determining gender. Females have 2 X chromosomes and males have only 1 X chromosome and what's called a Y chromosome. The X chromosome has other genes on it in addition to the genes that code for sex or gender. So the X chromosome and the Y, these sex chromosomes do contain some genes that are going to be responsible for some other traits or other characteristics. Their main trait or characteristic they help determine is gender. But going along with this sort of theme, the fact is that there are some other genes other than that for gender or sex that are held on the sex chromosomes. Mainly the X chromosome is the one that we're going to kind of focus in on, because that X chromosome, the 2 genotypes, if you will, male versus female, females have 2 copies of the X chromosome, and males only have 1 copy of the X chromosome. So here's a human male or female versus male karyotype, right? And here's those, there's those 22 autosomal pairs, right? There's that 23rd pair, right? That the sex chromosomes that determine gender. 2 X chromosomes in the case of females, and then males X and a Y. And you can see that the X chromosome is physically larger, right, than that of the Y chromosome. And this X chromosome carries information on it that determines gender, but it also carries some information about other traits on it. And a couple in particular that we're going to talk about starting right now. So the first is color blindness. So I don't know if any of you guys know of anybody that's colorblind, or maybe some of you might be actually colorblind. It's one of those traits or characteristics. It's certainly not debilitating or anything like that. It's just one of those things where color blindness is present within the human population. My father-in-law is colorblind, and mainly color blindness usually falls in the not being able to see like reds and greens very well. Now, the fact is that going back to this slide, in the case of sex-linked traits, it is more prevalent to be seeing these different characteristics in males because males only carry one X chromosome. So one example with color blindness, right? It's very rare to ever see a female that is colorblind. It's much more common to see males that are colorblind because females have two X chromosomes. And they need to have two copies of the quote-unquote defective allele or the color blindness allele for them to actually exhibit color blindness. So females are sort of most often, if color blindness is something that runs in a family, females are most often going to be carriers. Males are more prevalent to be to end up having color blindness because they only have one X chromosome. So here's an example of a couple. The parents were the female is a carrier for color blindness. She possesses a normal allele, and then she possesses a defective allele. The man, he has normal vision on his one X chromosome, so he has the normal allele. Now, when we do a Punnett square and cross these, the four possible outcomes, again, 50-50, it could be either male or female. In this case, the possible offspring, if they have a girl, it's either going to be normal vision with having both of the normal alleles, or normal vision but possessing or just being a carrier. So in this case, it's impossible for them to have a daughter that would be colorblind. Okay? On the other hand, if they have a boy, right, the possible outcomes if they have a boy would be that the boy would either have normal vision or be colorblind. Okay? Because again, it only is going to have received one of the copies of the defective allele to end up being colorblind. Okay? So another sort of look at this, okay? And to give you sort of an idea, the only way that you could ever have colorblind females or daughters, if you will, in this case would be if the female is a carrier for color blindness and the male is colorblind himself. And in this case, there is a possibility to have a female that would be colorblind. Right? So here in this spot, right, female genotype having two X chromosomes. B, the lowercase b represents the defective allele, if you will. Okay? In this case, you would have two copies of the defective allele causing for a colorblind female. In the other case, though, again, it's only 50-50, right? It's a 50-50 shot if they have a daughter that would end up being colorblind. But again, in this case, the daughter would be normal vision but a carrier. On the male side of things, on the bottom part of this Punnett square, right? Again, male is XY. Right? It'd be a 50-50 chance of having a male that's either colorblind, again, defective allele, or not colorblind. So now keep in mind, right, like if a colorblind female had kids with a colorblind male, then again, 100% offspring would be colorblind. Another example would be hemophilia. Okay? Again, hemophilia is a blood clotting disorder. Again, hemophilia, because it's a sex-linked trait, is more common in that of males than that of females. So here again, we have, a male, right, that does not carry the defective gene or defective allele. Lowercase letters represent the defective allele for hemophilia. Uppercase represents the non-defective form of the correct for the correctly working form. On the female side here, so female is a carrier. She has one of the defective alleles. Again, she's not hemophiliac. She's not affected with this disorder, but she's a carrier. So now when we do the cross, right, on the female side of things, right, for the female outcomes, it's impossible for them to end up having a daughter that would have hemophilia. The only thing is 50-50 chance that they would have a daughter that would be a carrier. But again, noticing that there's no alleles on the Y chromosome, and males only need to inherit one copy of the defective allele. Therefore, on the male side of things with the outcomes, right, it's a 50-50 chance that the offspring would be affected with hemophilia because males only need to inherit one copy of the allele. Next on the list, codominance. So codominance is when both alleles are expressed basically equally. So hence the word sort of codominance. It's not classic Mendel dominant versus recessive where the dominant form of the allele masks. We kind of use that word masks, right? The recessive form of the allele. In this relationship, it says to proteins and in relationship to proteins and enzyme production, enzymes that produce the phenotypic traits are found in equal proportions. So basically, what does that mean? Like these genes that we're talking about that have different forms of the alleles are going to determine ultimately what types of proteins are going to produce, that cells will produce to determine certain phenotypic outcomes. So like in class, we talked a lot about like this idea of like purple colored flowers versus white colored flowers in the Mendel examples. In the case where purple dominated over white as far as the dominant allele versus recessive allele, if that offspring had at least one copy of the dominant form of the allele, that meant that the flower colors were going to be purple, meaning that it triggered cells in that plant to produce a pigment color that would give the purple appearance or the purple pigment in production of proteins or pigments and proteins in the flower itself. So it was either purple or white. Now, in the case of codominance, though, again, you're going to look at the heterozygous case. So that's again, so like the heterozygote case would be, just to kind of remind you guys, so like, this is the homozygous dominant, capital letters, right? Sorry, that's a capital letter. And then homozygous recessive, okay, would be having both of the lowercase letters, but the heterozygote case, it has one copy of the dominant allele and one copy of the recessive allele. Okay. So in the heterozygote case, in those individuals that are heterozygous, in the case of codominance, what you see is these two distinct phenotypes are both expressed and they're both equally expressed. So interestingly enough. So let's go to an example. Let me clear all drawings here. Shut this down. Oops. And then we'll go to the next screen. So an example is like blood type. I don't know if you guys have heard of this, but like type A and type B and type AB blood types. Okay. Maybe some of you know what your blood type is. So those individuals that have type A will produce the type A antigen. Those individuals that have type B will produce the type B antigen. But those individuals that are type AB, right, will produce these antigens. And what's an antigen? It's basically a compound that will produce within our blood that prevents other forms of red blood cells, other attacking agents. It could be bacteria, it could be viruses, from ultimately inhabiting our blood supply. So these antigens are produced within our blood to be able to fight off other infections from actually invading our blood. So antigens A and B are produced in those that have type AB in equal amounts. So those that are type A, they produce only A antigens. Those that are type B will only produce B antigens. Those that are type AB will express both in equal amounts. Okay? So this is just a graphic to kind of give you sort of an idea what I mean by this. Right? So type A, the surface antigen is sort of the circle shape. They produce these antibodies that will attack and prevent any sort of type B. Type B individuals, the surface antigen, it's sort of, again, it's sort of this identifying tag on the surface of the red blood cell. They represent it with these little blue triangles. They make type A or antibody A antibodies that would prevent type A blood from being transfused, say, to a type B person. But that of a type AB person, they do not produce any antibodies. So a type AB person would be able to be transfused with either blood type. We'll talk a little bit more about this tomorrow in class. This gets a little bit technical, and I want to be able to draw some things up in class with you guys. I think a little bit easier sort of idea here, or a little bit easier expression, this is codominance that is expressed like in cattle. So another example here. So genotype of red hair versus white hair in cattle hair color. So here's a cross where we have two heterozygote individuals, right, that are crossing, and the resulting potential genotypes would be as follows, right? Having inherited two of the dominant forms of the allele, that cattle, okay, or that offspring will have only red hair. Those that were 1 out of 4 will have white hair, so both recessive forms of the allele, okay, would result in white hair. But here's the heterozygote case. Remember again, right? Anytime that individual inherits or is the heterozygote individual, it's both red and white hairs are being expressed or grown in equal amounts. You say hi to the class, Sanders?
Hi.
So codominance, anytime we have the heterozygote individual having a copy of the dominant allele and a copy of the recessive allele. Remember, in this case, in classic Mendelian genetics, it would state that having just one copy of the dominant allele would mean that it would show the dominant form of the trait. Okay? The only way to show the recessive form of the trait was you had to have 2 copies of the recessive allele. But in the case of codominance, having one of each results in both of the forms of the trait being expressed in equal amounts. It's kind of cool. All right, let me clear that. And we'll go to the next. Now, another sort of variation and related to that of codominance in some ways, but it's different. Right? So these are the two that you got to be careful about not getting mixed up. Is codominance and incomplete dominance. So in incomplete dominance, the dominant allele does not completely mask the recessive allele. So remember, these genes that organisms possess code for protein production. Ultimately, in the case where the heterozygote individual is exhibiting what form of the trait are they going to exhibit, they're going to end up exhibiting what appears as a blending between the 2 homozygous phenotypes. So meaning it's not producing one or the other, okay? It's not producing, in the case of codominance, both, but it's producing proteins and, say, pigments, in the example that we'll talk about in just a second, at a lesser rate, causing sort of an intermediate or blend to appear. So a type of plant that will show this are these plants called snapdragons. They're a type of flower, right? And in the case of the heterozygote individual, if the individual has both dominant alleles, the flower color would be red. If the individual has both recessive alleles, the flower color will be white. Okay? But if it's the heterozygote case right here in the middle, where it has one copy of the dominant, one copy of the recessive allele, the enzyme responsible for production of color is produced in smaller amounts than in those that for red. So that red pigment that produces red flowers is produced in high amounts in that of the plant that has both dominant alleles. The plant that has both recessive alleles, it's the lack of protein production, complete lack thereof, so it appears as white flowers. But in the case right in the middle here, right, it produces those proteins at a lesser rate, those protein pigments at a lesser rate, so the color of the flowers appear as pink. So pink is kind of a mix, if you will, or a blend between red and white. Okay, so let me here clear these drawings. Let's go to the next slide and take a look at incomplete dominance. It's a little visual here, right? So here's that same cross. So let's say we take two pink snapdragons, right? And we cross those two pink snapdragons, right? So here's my Punnett square. The resulting outcomes, right? I can't get this to draw anymore. The resulting outcomes would be homozygous dominant, heterozygous, and homozygous recessive. The heterozygote case, again, will produce pink flowers by comparison. Homozygous dominant, only red. Homozygous recessive, having two copies of the recessive allele, produces no red pigment at all. Heterozygote case, it's a blend between the pink and the red here. Okay. Another example of incomplete dominance in that of like humans is lactose intolerance. Maybe you know of somebody that's lactose intolerant. Maybe you know somebody in our classes are lactose intolerant. But there's also lactose sensitive. Okay? So there are, again, the heterozygote individuals. So capital L represents the dominant form of the allele. Okay? Lowercase l represents the recessive form of the allele. Right? So if we're crossing two individuals that are lactose sensitive, okay, a person with lactose intolerance lacks the enzymes to digest milk. So some people can have some milk but do not digest lactose as well. So here's sort of the deal. If I could get my, it's not doing this for me. All right, no big deal. So the homozygous dominant example, having both of the dominant alleles, those individuals can digest lactose normally, okay, or lactase normally. Okay? Again, lactase is the form of sugar that's in dairy products. Those that have inherited both of the recessive alleles, okay, would not be able to digest dairy products and lactose in dairy products at all. So consequently, it can lead to some digestion issues. So they have to avoid dairy at all costs, sort of. Okay? And then those that are caught in the middle, again, the heterozygote individuals, having the dominant form and the recessive form, one of each allele. They're lactose, not lactose intolerant, but they're lactose sensitive, meaning that they can digest some dairy products, some milk, but they can't digest it as well as those that are normal. Okay? So again, it's not quite as extreme as those that are lactose intolerant. Okay? They maybe have to watch their dairy intake. They can't have maybe large amounts, but some of it's not really a big deal. Okay. All right, so we'll stop it at that for now. Again, tomorrow I'll kind of buzz through these sort of three things that we looked at, and then we'll move into polygenic traits tomorrow, pleiotropy, and genes in the environment. I would suggest that, maybe just for the interest of time tomorrow, you guys do some reading ahead within this presentation and look at polygenic traits, look at pleiotropy, and look at genes in the environment, just so you've sort of read through it, and then we can move through the rest of this tomorrow in class. Don't forget, again, tomorrow, 2 assignments that are due: corn activity and genetics packet number 1. Come with questions that you might have, especially on genetics packet number 1, if you want to talk about those last minute. All right, that's it, guys, for today. We'll see you tomorrow.